Hi, I am Yuxuanqing. Today I'm going to talk about the REM sleep muscle paralysis and how its dysfunction can lead to the REM sleep behavior disorder. First of all, what is the REM sleep muscle paralysis? It is characterized by continuous sustained absence of muscle tone, which is also known as REM sleep atonia, and a few intermittent bursts of muscular activity that can be associated with minimal muscle twitches, resulting in the brief and small distal movements of the limbs, ears, orofacial area, jaw, and tail, which is known as the REM sleep EMG phasic activity. So, the REM sleep muscle paralysis requires sustained inhibition throughout the REM sleep period of the tonic EMG activity that underlies the postural tone and disabilitation of the phasic EMG activity that influence the movement and locomotion. In other words, muscle tone in REM sleep is reduced by combined activation of inhibitory system and inactivation of facilitatory system. The final result is the inhibition of the motor neurons located in the nuclei of the cranial nerves, of the brainstem like hypoglossal facial, and of the motor neurons of the spinal cord, both resulting in the skeletal muscle atonia and minimal intermittent muscle twitches. By applying the cytotoxin injection to damage specific brainstem nucleus, researchers found that the critical structures for REM sleep generation and REM muscle atonia are located in the mesopotame tegmentum and in the ventromedial medulla. In these regions, there exist a large number of nuclei that send projections inhibiting or activating the nearby nuclei in the brainstem or other distant brain regions. Yet, what nuclei in the mesopotame tegmentum and VMM? In the mesopotame tegmentum, the key structure is the ventral region of the sublateral dorsal tegmentum nucleus, the VSLD in rodents, which is equivalent to the subcutaneous nucleus in human. And in the VMM, the RNG, GIA, and so on promote the REM sleep paralysis. The VSLD generates muscle paralysis by two different pathways. There is a directed pathway of excitatory VSLD glutamatergic projections to the interneurons of the spinal cord, which in turn inhibit the ventral horn skeletal motor neurons with GABA or glycinergic input. An indirect polysynaptic pathway consists of other descending glutamatergic VSLD neurons that send their projections to the VMM, which in turn inhibits the motor neurons of the ventral horn of the spinal cord, resulting in muscle paralysis. The neurons of the VMM release GABA or glycine to the ventral horn motor neurons and glutamate to the spinal cord, the GABA glycinergic interneurons, which in turn inhibit the motor neurons of the ventral horn. The red nucleus in the midbrain activates the motor neuron in the spinal cord and produces the muscular twitches during the REM sleep. The VMM inhibits the activity of the red nucleus to reduce the production of the phasic EMG activity that could be excessive to allow the release of the movement. Damage to the key structures that generate REM sleep paralysis could lead to the REM sleep behavior disorder, which is also called RBD. In humans, RBD is clinically characterized by unpleasant dream recall, abnormal simple and elaborated behaviors during the REM sleep, the impairment of glutamatergic neurotransmission from the subcutaneous nucleus, and or the GABAergic and glycinergic neurotransmission from the VMM underlies the pathophysiology of RBD. Alternatively, it is plausible that RBD can also be caused by damage of the anatomical connections of the subcutaneous nucleus and VMM, with other structures such as the dorsolateral hypothalamus and the limbic system. Some sleep disorder, like RBD, is often the initial manifestation of devastating neurodegenerative diseases, like Parkinson's diseases, that carries important diagnostic implications that opens a window for neuroprotection. And that's the end, and thanks for watching. Here's my reference list.